So today we are going to show you some videos and we will perform the actual demonstration. We prepared this video, we the NIV team basically prepared these videos for you. So first, this is basically the method of RNA extraction or the nucleic acid extraction through the automation by using this kit. Over here you can see this D12 plate. Yesterday you haven't seen this Vakat. Have you shown this D12 plate?
Yes.
So no?
Yes.
Yes. Okay, okay. Today, we will brief you about this D12 plate. In this D12 plate, there are designated different rods for the lysis buffer, for the washing buffer, for washing buffer 1, washing buffer 2, and then eventually the illusion of RNA or DNA, or the nucleic acid. So initially, over here, we are adding proteinase K. Why proteinase K or the carrier RNA? For lysis of protein.
Yes.
This carrier RNA is for lysis of protein. So what is inside the lysis buffer? EDTA and okay, just forget about this plate. Okay. If you are doing manual RNA extraction or DNA extraction, you use first the lysis buffer. So what is the purpose of that lysis buffer? Of cells. To disrupt the cells. Yes. Okay.
Breakdown.
And the lysis buffer contains different detergents, EDTA, and different other things. And that we add the carrier RNA. Why carrier RNA? Why this proteinase K? What is the purpose of this carrier RNA? To it will bind to what? Basically proteinase K or carrier RNA. Carrier RNA, usually in manual extraction, we are using this carrier RNA because it supports our RNA. Or proteinase K, it basically remove all types of different proteinases. Okay, eventually, what is the purpose? Support the RNA. So, this is the program. We will show you this machine, which is 96-well RNA extractor. By using this machine, we can extract out 96 samples in 35 minutes. Okay, so during COVID, we use this machine for diagnostics. You can see French.
Yes.
English and French both. One deep well plate with the lysis buffer, then another with the washing one, washing two, next one or the final one with the illusion buffer. Okay?
One is washing buffer and other is lysis buffer and finally
Lysis buffer, washing buffer one, washing buffer two, and then final illusion. And in last, we have our sample in this deep well or RNA in this deep well phase. After doing experiment with the like SARS-CoV-2, this is the doping procedure. C. Sanitizing.
This one is good. This one is good.
This is the face shield. So these two videos are the most relevant one for today's lectures, because after that we will go for the demonstration of RNA extraction. So do you want to perform the manual extraction or the automation?
Manual.
Manual, right? Why manual?
We don't have to see it. Yes. Yes. There is manual automatic. This automation can be sometimes it can give us error. If we know the manual method, we can solve that error and we can make the troubleshooting solving.
Okay, what will you do when there is an outbreak and you have a bulk of samples like 2,000, 3,000, 5,000 samples per day? So you move towards the manual one or for the automation?
Both and you.
And about the error rate, if we are using the different types of control, we can nullify the errors in the mesh automation as well. So today we will perform the automation of two different kinds of automation machines for the nucleic acid extraction, and we will also demonstrate about the point of care. So do you have any idea what are the point of care diagnostic kits? Yes, Samira.
Point of care diagnostic kits are those kits which are easily like because there are urinary kits or pregnancy kits. Yes, similarly, there are different chromatographic based ICC that can be used for detection of antigens.
We can call it rapid.
Rapid test, rapid kits. So we will demonstrate you all about the point of care kit, like the lamp assay, because like in some areas and in like developing countries, there are very limitation for the PCRs. So for the lamp assay, we can use heat block for the diagnosis. So we will perform point of care assays, and under equipments, Dr. Sharmeen will explain about the various scan, which is a sort of a spectrophotometer, but we can take luminescence, absorbance, chemiluminescence, everything with a range of 100 to 1,000 wavelength of spectrophotometer, and then we will perform the cell counting by the cell counter, not by the reward chamber, by using the cell count. Okay. So next we will have our lecture on antivirals by Dr. Shumaila Osman. She will be online in 2 minutes. So meanwhile, you can take tea.
Antivirus.
And
Shorting.
Dr. Shumaila Aman. Video. Yeah. Okay. Tell me the difference between carrier RNA and protein. What is carrier RNA? When doing extraction, nucleic acid extraction, one step is the addition of carrier RNA. So, what is the purpose of that carrier? Anybody else?
I don't know if it's carrier.
No. Anybody else?
Proteases, this protein by the help of.
Basically, proteinase, kinase break down all the proteinases like nucleases in order to support your RNA. And what is carrier RNA? It's covered, extracted RNA. Yeah, it is. We will basically explain it in deep, inshallah, when we will perform our experiment. Okay. वालेकुम अस्सलाम, आपने लिंक देख लिया? हाँ ठीक है, सही है, बस फिर आ जाइए। ठीक है, ब्लर करना है। ठीक ठीक। जी 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 जी। So today we have Dr. Shumaila Aman. She is pharmacological sciences expert, and she has an extensive experience in molecular medicine and experimental pharmacology, currently serving as an assistant professor at Dow College of Pharmacy, Dow University of Health Sciences, Karachi, Pakistan. She holds a PhD degree in pharmacology and completed a postdoctoral fellowship at Hunan University of Chinese Medicine, China, under the Talented Young Scientist Program awarded by Ministry of Science and Technology, China. Her expertise encompasses biochemical, molecular, and computational pharmacology, supported by multiple publications in international index journals. A reviewer for Journal of Ethnopharmacology, Phytomedicine, and Natural Product Research, and a member of American Heart Association, British Pharmacological Society, and International Society on Thrombosis. And she remains dedicated to advancing translational pharmacological research and mentoring the next generation of scientists. We welcome Dr. Shumaila Aman. Dr. Shumaila, can you please unmute your mic?
Hello, Assalamu alaikum. Can you hear me and can you see the presentation as well as my screen? Hello.
Yes, ma'am.
Okay. Thank you. Thank you so much, Dr. Sabah, for such a generous introduction. And I welcome everybody on this great forum of the training course in virology and pandemic preparedness. Um, First, I welcome you to my session, which is about the antiviral drugs, which is particularly emphasizing the most commonly having viral infections which are occurring these days, especially in the pandemic condition. The importance of this lecture is about the preparedness of the basic treatments which we will having in the diagnosis as well as treating the common viral infections. So I start my presentation, which comprises of mechanism of action and pharmacological insights of the main treatments which are used for the viral actions as an antiviral drugs. Okay. Before formally coming to the drug therapies, we will first look into the brief life cycle of the virus. As we already know, the virus is comprises of the structure which is comprising of the outer core and some which comprises of the outer core having the proteins. These proteins are of different types: glycoproteins, GP120, GP40, which are normally called as the protein spikes. These protein spikes are used for the attachment of these viral structures to the host cell. In this picture, you are looking at the influenza virus, which is a virus having the host cells from the animal source, human, as well as you can see the bacteriophage, which is virus in which the host cell will be of the bacteria. Among these two structure, the outer core and the inner part, which is comprising of either the RNA or DNA, which is a genomic structure of the virus. So these are two different type of structures, and as well as we have some complex structured viruses also. Similarly, the DNA nuclear material comprises of either the RNA or DNA of the inside the virus. Normally, when a virus and the total life cycle of the virus is like this, the first step is the attachment of the virus to the host cell. In this picture, you can see the host cell is the bacterial cell. The virus will bind itself from its glycoproteins to the cell surface. Then through the endocytosis, it releases its genetic material into the host cell. Then inside the host cell, these are the virions, which are comprising of the complete structure of the virus, which is called as the fourth step maturation. And finally, they will release the endothelial or the bacterial cell releases these viruses into the body of the organism. Antiviral drugs will going to stop, inhibit, or attack at different stages of these life cycle of the virus. So the understanding of the structure of the virus and the life cycle of the virus is very crucial, because if we say the infection is very spreading, you can say like influenza virus, which is very fast growing, fast spreading infection. In that condition, the drug of choice for that type of infection is something different. And if we say the infection is very latent type, that is the viral will going to stay inside the body, and when our immune system will get suppressed, then it will become again activated and then attack the human body or the bacteria, whichever it is. This picture shows the cell cycle of an influenza virus. In this condition, the host is the human being. The same different steps will be followed in this human body type, similar like we have seen in the bacteria. The first step is of attachment of the influenza virus to the human epithelial cells. This attachment is via the glycoproteins. So we can say the first drug of choice which will inhibit this process can be the inhibition of this attachment phase, in which the drug will stop the glycoprotein to get attached to the epithelia or cell surface. Then we have second phases of penetration. In this penetration, the viral cell will completely entered into the host cell, and in the third step, there will be uncoating of the caspid of the virus. This uncoating will going to release its nuclear material and every type of material in the human cell. Then this, which is the sixth step, release. New viral particles are made and released into the extracellular fluid. Then the fifth step is assembly of the new virion, new phages, which will inside the epithelia. And finally, the biosynthesis, in which the human cells nucleus will going to release the genomic either RNA or DNA material of the virus into the cytoplasm, which will incorporate to form a new complete daughter cells or virion or the phages. So these steps are is similar in the bacteria or human body, and they are very crucial to learn about, because at each and every step we can stop the inhibition of the growth of this virus. Here I will make emphasis on another point, that there are some kind of infections which are active at due time, and then when we treat them, they will get subside and completely eradicate from our body. However, there are some infections which will become latent in our body. Latent means they are present inside the body, but in their latent or inactive form. For example, herpes simplex virus, which become latent in our brain parts, or you can say in the neck region, in the lower part of the brain. They will reside as a latent infection, and when there is immunocompromised conditions, either diseases or nutrient lack, then these become active again and attack the human. So up till now, whatever the drug we have, they are active, they are good for treatment during the active phases of bacteria. And by active phases means these six steps which you are looking at, the attachment, penetration, uncoating, biosynthesis, assembly, and finally the release of the material, the release of the virion. So these are the active steps in which these drugs can be can be used for the treatment purposes. Very quickly, we go through different viral types which are having in our environment very commonly. Herpes simplex virus 1 and 2, HSV 1 and 2. HSV 1 is called as cold sore viruses, while HSV 2 are particularly for genital herpes virus and cause the genital infections. HVZ virus, varicella zoster virus, cause the chicken poxes and the shingles. cytomegalovirus, Epstein-Barr virus, human herpes virus, human papilloma virus. These are the major common virus. And another broad category is human immunodeficiency or HIV virus, which is the retrovirus, which we will study in the detail, as well as the COVID coronavirus, which we will look separately. Now coming to our main theme, that is the antiviral drugs. Antiviral drugs are the due time requirement need of our time so that we can have new discoveries and we can gain the treatment strategies, new treatment strategies against the upcoming pandemic situations. The normal or conservative conventional therapies, including the antiviral drugs, which are purine or pyrimidine analogs, which are the nuclear material, and these drugs are particularly the prodrugs. The prodrug is the terminology which is used for that they must become activated when enter into the body. So in our antiviral conditions, antiviral drug, they must be phosphorylated by the viral or cellular enzymes in order to become active, and then they will treat the disease. The good thing is these drugs are activated usually mostly by the viral enzymes which are present within the virus. So that means if the virus is present and it has that enzyme in our body, entered in our body, only then these drugs will become activated. And when they will become activated, they will only attack these cells in which that enzyme will be present. That means they are not damaging to our normal human body cells. Only those cells which are capable to contain those viruses as well as those enzymes. So this is a good strategy to not damage the normal human body cells. Acha. Antiviral agents inhibit active replication, so the viral growth resumes after the drug removal. When we remove the drug, then again, these activated forms are present in the body. This is more like the pharmacokinetics. That is, when we take the drug, it will become activated, and the inactive conjoint of that drug will get release out from the body, but in our body the active part will be there and cause the actions. Current antiviral agents do not eliminate non-replicating or the latent viruses. They are not effective against the latent virus, as I already have explained. Effective host human responses remain essential for the recovery from the viral infection. This is the most important point. If our immune system is not very active in both the situation, that is before the infection and after the infection, the even the treatment is very particular. Even after that, the disease will become recurrence. The recurrence of the disease will be there again. Clinical efficacy of these drugs specifically depends on achieving the inhibitory concentration at the site of infection in the infected cells. That means the drug, when enters into the body, it should get to the infected area where the virus has infected the organ. Like in cases of hepatitis C virus, we have given the drugs, so it is particularly important that it should enter into the liver as well as it get the therapeutic levels within the liver, so we can have these effects of the antiviral therapies. If we see the classification of the drugs, antiviral drugs, we have those drugs which will attack the viral DNA, and these are particularly effective in herpes simplex virus and CMV, that is the cytomegalovirus. Then we have those drugs which will inhibit the RNA virus formation, specifically anti-influenza drugs, herpes simplex and HIV antiretroviral drugs. Then we have reverse transcriptase inhibitors, which are the enzyme inhibitors, and reverse transcriptase role is in the synthesis of the nuclear material of the virus. Then we have protease inhibitors, again the enzyme inhibitors. Then we have fusion and entry inhibitors, which is the first step that viral will come to the host cell and it attaches itself to the endothelia or the cell membrane of the cell, either the bacteria or the human. And the second step is the entry of that virus into the epithelial or endothelial cells. Then we have nucleoside and nucleotide analogs. They work as the copy material of the viral RNA, and then they will get inserted into the viral RNA and thus stop the synthesis of normal viral RNA or the DNA. And then we have non-nucleoside polymerase inhibitors. These are again attacking the synthesis of the nuclear material by inhibiting the enzyme polymerases. Along with these, there are many other classes which will either become not very active or they are not very much used since we have better effective agents. So we will not study here about them. General mechanism of all the antiviral drugs, including the different steps of the viral life cycle. Firstly, they will either block the viral attachment or entry of the virus into the cell. Secondly, they inhibit the uncoding of viral genome. When viral, if enters into the cell, then there will be the cell uncoating of the viral. Then it will release that material into the cytoplasm. Third step is inhibiting the nucleic acid synthesis of the virus. Then we have inhibiting viral protein synthesis or processing into the new virion. And finally, the blocking the viral assembly or its release outside the host cells. So these are the different steps on which we can act by these antiviral drugs, and they will going to inhibit the release of the virus into the human bodies, thus they can attack some other human cells. We have anti-herpes virus agents, for example, acyclovir, valacyclovir, famciclovir, genicyclovir, trifluridine, vidarabine, etc. Actually, we have multiple drugs that can be effective to treat any infection. In this scenario, if we give example of anti-herpes virus agents, we have variety of drugs. Then what is the condition and how we should choose that drug? This depends on the stage of the disease. If the disease is at the initial stage or disease is at very fast growing stage, fast spreading condition, then we have to then we should look into which type of the drug we should choose at that time. The first one, we have nucleoside and nucleotide analogs. These are the copy of the nucleoside material of the viral DNA. They will get inserted into the viral RNA or DNA and cause the inhibition of that nucleoside nucleotide generation materials. They require activation by the viral or host kinases. Here you have looking into two words. Either the kinases which belong to the virus or kinases belong to the host. Viral kinases usually involve thymidine kinases which are present in the virus, and when the drug will interact with those kinases, then it will become active immediately and then attack that virus. Similarly, some drugs will be activated by the host kinases as well. So that means these drugs, when entered into the body, they must become activated by utilizing these kinases in order to perform their action. These drugs include acyclovir, genicyclovir, zidovudine, and tenofovir. These drugs are conventional drugs. We have been using them for so long time, and still they are the best choice in the initial or mild stages of the infection. Valacyclovir is a prodrug of acyclovir which has the better bioavailability. Again, I am repeating the prodrug, and the prodrug is the word which means that it should become active and then it will convert into its active form. Femicyclovir is the choice which is hydrolyzed to another compound which is called as pencyclovir. And why it is necessary for its conversion to pencyclovir? Because pencyclovir has greater bioavailability. That means it will enter into the body and most of its form will get activated and act on the human or viral infections for the treatment as compared to the parent compound, which is femcyclovir. Pencyclovir has the condition that it can be used only topically, while femcyclovir can be administered orally as well. The mode of action of these nucleoside and nucleotide, as you can see in this picture, these nucleoside analogues, valacyclovir converted into acyclovir and femcicyclovir converted into pencycyclovir, they will acted upon by the viral thymidine kinase UL23. Then they will forming the monophosphate, then utilizing the host kinases forming the diphosphate and triphosphate. And then they will incorporate it into the RNA or DNA material of the virus. Here in this example, you can see the DNA polymerases forming the DNA, and then here these drug agents will be incorporated at these different points of this synthesis and thus inhibiting the normal synthesis of the DNA. These all drugs are phosphorylated by viral thymidine kinases, and they are acting on actively replicating viruses. That means they will be very effective in the very fast growing virus or spreading virus. Acyclovir is a good choice of drug for multiple infections. It belongs to guanine nucleoside analog and it has the mechanism of action activated by viral thymidine kinases and thus inhibiting the DNA polymerases. It is very specific for herpes zoster and varicella zoster viruses, which are commonly called as shingles, which are the kind of rashes over the body. Its pharmacokinetics has low oral bioavailability and it is eliminated from the renal area. And adverse effect are nephrotoxicity and rarely the neurotoxicity. Then we have the second choice of drug, which is gencyclovir and valganciclovir. And it is specific for cytomegalovirus therapy. Again, these drugs are activated by thymidine kinases or UL97 phosphotransperases into first active form and then second active form, and further utilizing the cellular humans host cellular kinases, they will finally become the activated drug. Their mechanism is similar to acyclovir, but only the difference is they are activated by the UL97 kinases, and their toxicities include bone marrow bone marrow suppression. Valganciclovir is again a pro-drug of gencyclovir, so its oral bioavailability is now becoming better. Here we should emphasize or we have to understand this point that continuously, although we are having a good agent like acyclovir, we are continuously into the research to get the newer compounds which have more bioavailability. And what is the purpose of this more getting more bioavailability? That if we have more active form inside the body at lower doses, then we have the lesser side effects of that particular agent. So this is a successful strategy to synthesize new type of compounds from originally conventionally available drugs, because in that way we can get the active, we know the mechanism of action of that compound, we know that it is already it is already an active form of drug, and we can increase its bioavailability by forming its more different medicinal applying medicinal chemistry and forming the more bioavailable forms. Similarly, other drugs like foscarnet and cidofovir are also those nucleoside or nucleotide inhibitors. They are directly inhibitor of DNA polymerases, and for them there will be no activation required by the kinases. Cidofovir required the host kinase activation. They are active against cytomegalovirus, and nephrotoxicity is the major adverse effect. Nephrotoxicity is the renal toxicity, and we have to adjust the dose depends upon the creatinine clearance of the human body. Antiviral spectrum is mainly it is major common infections. All these drugs will have the infection, and the most successful drug is the acyclovir, which is also available as an oral form. The resistance. Nowadays, the one of the major problem you can say is the resistance against these microbes. Resistant strains are those viral strains. They can have different mode of actions. The first one is that these strains can lack the thymidine kinase enzyme. These enzyme which is involved in the activation or phosphorylation of many drugs like acyclovir. So if the virus have no thymidine kinases, then we have no option to give the acyclovir. Some of these clinical isolates are resistant on the basis of thymidine kinases as well as some other kinases. And some of these agents do not require activation by viral thymidine kinases, and thus we can use them in such condition of the resistant species in which there are no kinases. For example, we can give foscarnet, cidofovir, and trifluridine in resistant cases of the virus. Gencyclovir can also be used as some infections in which it is not commonly used because we have acyclovir. But if there is resistance against the acyclovir, then we can have gencyclovir as well, which is a very good and effective agent. Adverse effect can be myelosuppression, which is actually the bone marrow suppression, nausea, vomiting, diarrheas, neuropathies, and some CNS effects and the hepatotoxicity. One thing I will emphasis here is that most of these antiviral drugs have the effects on our metabolism, that is through the cytochrome enzymes. So if the cytochrome enzymes are altered by these drugs, then we have to check the dose of these agents and some other agents as well, which can be given as the concomitant drugs or the helper drugs. Then the second most common infection we have is the antiretroviral therapies. They are again comprises of the different classes like NRTIs, NNRTIs, protease inhibitor, integrase inhibitor, or interior fusion inhibitor. The most important thing is the last point, which is the combination therapy called as HAART, heart therapy, which including the those resistant strains which will effective against those resistant strains when we give these combination. That means we are giving those drugs which are acting on the fusion or entry phases. Plus, we are giving those drugs which are acting on RNA or DNA material incorporation or their termination of the release, so that we can attack the different stages of the cycle and we can suppress the infection. So among antiretroviral agent, different classes. The first one are reverse transcriptase inhibitors. In this picture, you can see when HIV-1 virus enters into the human cells, it incorporated inside the cell and start to replicate its DNA material and involving the reverse transcriptase enzyme. These drugs, which are in our specific nucleoside analogs of reverse transcriptase inhibitor, they will bind to this reverse transcriptase and stop the this chain reaction and thus inhibiting the synthesis of this viral DNA material, and in this way they will stop the function of the stop the phages synthesis. These the example includes zidovudine, lamivudine, abacavir, which are very successful drugs, and in NRTIs we have efavirenz and nevirapine. They require activation by our human enzymes. This is the phosphorylation, mono-di, and then triphosphorylation. They inhibit the HIV-1 reverse transcriptase enzymes incorporated into the DNA and inhibit the DNA viral. And they are active against HIV-1 as well as HIV-2. Then we have second class, which is protease and integrase inhibitors. These inhibitors cause the cleavage of GAG-Pol polyprotein. GAG-Pol polyprotein is actually, GAG is the word for structural proteins which are involved in the structure synthesis of the virus, and Pol are the polymerases. These two are the couple proteins which work in the growth of the virus. So these GAG-Pol proteins are inhibited by the drug protease inhibitor like reltigravir and dolutegravir. These enzyme in this picture you can see these enzyme will stop these drugs will bind to the such polymerases and inhibit the viral DNA synthesis. Then we have entry and fusion inhibitor drugs. The mechanism of these drug is very versatile. As you can see, the first step, when the CD4 binding happens, the upper part is the viral structure and the cell membrane is the host cell membrane. This virus will come near to the cell membrane and the host cell CD4 will bind to the viral DNA material. Initially, the GP120, which is glycoprotein 120, will bind to the CD4 receptor of the host cell, and then conformational change happen at the V3 loop of the virus. Then this GP41 material conformational change happen, and it will going to bind to the cell membrane receptors. These drugs, which are entry and fusion inhibitor, work at different steps. That they can inhibit the CD4 receptor of the host cell. Secondly, they can act on the CCR5 inhibitor, which is the second step of the fusion of the viral virus to the host cell membrane, and CXCR4 inhibitors, which act on the binding of CCR5 and CXCR4 at the host cell membrane. Then we have respiratory viral infections. Commonly, we have influenza virus, amantadine and rimantadine are the most common drugs, and the respiratory syncytial virus, ribavirin virus. Anti-influenza drugs have the action on inhibiting the ion channels, which are M2 ion channels present on the viral DNA. These ion channels will be blocked by these drugs, amantadine, and then they will stop the fusion or entry of these viruses into the human cell. Second, we have neuraminidase inhibitor. They will stop the last step, which is the release of the virus into the human body. And then we have CAP-dependent endonuclease inhibitor, baloxavir, etc. These are the inhibition of the mRNA synthesis. So in this way, these three type of drugs acting at the initial step, that is the entry, the second step, that is the RNA biosynthesis, and the last step, release of the virion. So by combining these three drugs, we can inhibit the activity completely. Ribavirin is the drug of choice for many infections like bronchiolitis, pneumonias, and it is very effective drugs. You've been used nowadays. It causes the inhibition of RNA polymerase and the DNA material. Then we have anti-hepatitis C virus antiviral drugs. These are different drugs which can be protease inhibitor like simiprevir, polymerase inhibitors, specifically NS5B polymerase enzyme and NS5A polymerase enzymes. Or we can use the combining therapy of these drugs. The mechanism of action is same, like they will entered into the body, and when they entered into the human endothelial cells, and when they will entered in either the hepatocyte, there is a formation of virion assembly, and this assembly can be inhibited by these different agents at different polymerases. Then we have classes of interferons, which are again very effective against the hepatitis infections, as well as we can use some immunomodulators with them. Interferons are the natural protein produced by the immune systems, and they are divided into alpha, beta, and gamma, and they work by the produce only interferons are produced by the lymphocyte and natural killer cells, and they work by their immune regulating effects. Then we have anti-SARS-CoV-2 agents. These are the newer agents, for example, remdesivir, molnupiravir, and nirmatrelvir, which is commonly called as Paxlovid. Paxlovid is a very successful newer agent, inhibits main protease which is called as MPRO. If we look at the mechanism of the SARS-CoV-2, if we only look at the first step, that is the fusion of the virus to the upper membrane or cell membrane of the cell, we can see ACE2 cells, receptors, and these drugs are very effective against these ACE2 receptor. Similarly, when this virus entered into the human cell, there is involvement of some proteins, polymerases. These drugs can affect specifically these COV2 polymerases, and then they can inhibit the spread of this SARS-CoV-2 infection. Uh, as we have look into the profile of the resistance, which is mainly the resistance is due to some absence of phosphorylases or mutation of viral enzymes. So we have to see some newer agents against these resistant species, or we can give some combination therapy, but again, more the drug, more will be the side effects. So we prefer that we should give newer agent with lesser doses, with targeted precision therapy, that they will act directly to the cells which is required, at which they are required. And lastly, the limitation is due to their toxicity. All of these agents have major toxicities on the kidneys, nephrotoxicity on the liver, hepatotoxicity, metabolism, bone marrow suppressions, our immune cell suppressions, anemias, and some mild things like nausea, vomiting, etc. So these are some resistance and limitation against these antiviral therapies. And this we have already discussed about the COVID-19 treatment. The future direction is we have to develop broad spectrum antivirals which can have effectiveness against many type of viruses. We should look into the precision therapies or host directed therapies which will act directly to the specific human being requirements. Nanotechnology based delivery will be targeted as well as lesser the dose. And finally, the CRISPR mediated antiviral strategies are the most successful things, but obviously they are the most expensive as well. So the takeaway messages: antiviral act on distinct viral life cycle stages. Drug selection depends upon the type of virus as well as their resistance patterns. Combination therapies are very efficacious therapies, and thus we will prevent the resistance as well. And future antivirals should aim for higher specificity as well as the safety. So here I will conclude my lecture, and I hope in this short time, this brief introduction, just only a brief introduction to the drugs, antiviral drugs, will be very good for you in your upcoming training sessions. Thank you so much.
Thank you very much, Dr. Shumaila, for such an informative lecture. And if you have 5 minutes, we can take questions. Any
Obvious.
questions? Yes. Can you please come aside because पका ये चल नहीं रहा, बिल्कुल नहीं चल रहा।
First introduce yourself and then ask. Okay.
Good morning, doctor.
Thank you.
My name is
Good morning.
Ekanya Rilindis from Cameroon. I
Oh,
just
well.
have a question
Mm.
here
Mhm.
because you made mention that purine and pyrimidine analogs, they are the
Yes.
best suited antiviral drugs. So my question is, what about them that makes them best suited?
Okay. if you use the word best choice, it is not actually, we can say, best choice to single drug. We have many drugs which can be best choice. If we only say about the acyclovir, the good choice we have, because it has the good bioavailability. That means it will entered into the body and have reached the target organ, target cell to the body, and it has lesser side effect compared to those like gencyclovir or some other drugs, which will be having effects like bone marrow suppression. So when we say a good choice of drug or a best choice of drug, that means it has the good bioavailability, it has specificity more, that it will go towards its site of action, and it has lesser side effects. That makes any drug the best or good.
Okay, thank you. My second question is, what is the difference between influenza and para-influenza virus?
Influenza and paravirus influenza has usually the main difference is the site of action. The influenza virus attack the upper respiratory tract and lower respiratory tract, while the other viruses are more towards the lower respiratory tract infections.
Okay, thank you.
You're welcome.
Thank you very much, Dr. Shumaila.
Thank you so much, Dr. Sabha.
Okay.
Thank you.